Welcome to the Biology Lecture Standard 12 2020-21. The second chapter of Unit 1 is Reproduction in Lower and Higher Animals. Contents: various methods of reproduction in animals, the human reproductive system, gametogenesis and fertilization, early embryology, parturition, and reproductive health. Fertilization or Syngamy. Sexual reproduction primarily involves formation and fusion of gametes. Fertilization is the later process which involves fusion of the haploid male and female gametes, resulting in the formation of a diploid zygote, that is 2N. Like in other mammals, in humans, the process of fertilization is internal and it usually takes place in the ampulla of the fallopian or uterine tube. The fertilized egg or zygote will develop into an embryo, and this process occurs within the uterus. This is a diagram which is exhibiting the process of fertilization and zygote formation. Mechanism of fertilization. Semen released during ejaculation has sperms and some secretions. The coagulated semen now undergoes liquefaction and sperms become active. The mechanism of fertilization is as follows: A. Movement of sperm towards egg. B. Entry of sperm into the egg. C. Activation of A. Movement of sperm towards egg. It involves capacitation of sperms reaching the vagina. Here as many as 50% are demotilized or broken or destroyed. Remaining sperms undergo capacitation. This process requires 5 to 6 hours. Acrosome membrane becomes thin. Calcium ion enters the sperm and their tails begin to show rapid whiplash movements. As a result of capacitation, sperms become extra active and begin to start moving upwards from vagina to uterus and to the oviduct. The prostaglandins activate the sperm. The vestibular secretions of the female also enhance sperm's motility. The sperms swim at an average speed of 1.5 to 3.0 mm per minute. Sperms reach up to the ampulla as a result of their own swimming and partially by contraction of uterus and fallopian tubes, stimulated by oxytocin of female. After capacitation, the sperms may reach ampulla within 5 minutes. Sperms can remain viable for 24 to 48 hours, ovum for about 24 hours. B. Entry of sperm into the egg. Out of 200 to 400 million sperms, only few hundred manage to reach the ampulla. Though many sperms reach the ampulla, but only a single sperm fertilizes the ovum. A sperm after reaching the egg or ovum comes to lie against it. Its acrosome releases lysine, hyaluronidase and corona penetrating enzyme. They separate and dissolve the cells of corona radiata, so the sperm head passes through the zona pellucida of egg. The zona pellucida has fertilizing receptor proteins ZP3 and ZP2. The fertilizing binds to specific acid protein anti-fertilizing of sperm. It brings about attraction of sperm to the egg to enhance of fertilization. Fertilizing interaction is species specific, thus the fertilizing anti-fertilizing reaction is also called compatibility reaction. Acrosome reaction. As the sperm head touches the zona pellucida in the animal pole region, its acrosome covering ruptures to release lytic enzymes acrosin or zonalizin. They act on the zona pellucida at the point of contact. This causes egg reaction. A small fertilization cone or cone of reception is formed on the egg membrane. The sperm head comes in contact with this cone. It results in production of a weak wave of depolarization. Plasma membrane of the both cells dissolves at the point of contact. The sperm nucleus and the centrioles enter the egg while other parts remain outside. As soon as the sperm head touches the vitelline membrane, a cortical reaction gets activated, changing the vitelline membrane into a fertilization membrane by deactivating the sperm receptors of zona pellucida. A distinct perivitelline space is created around the fertilization membrane. This prevents any further entry of other sperms into the egg, that is, polyspermy is avoided. C. Activation of ovum. The ovum before fertilization was at metaphase 2 stage. With a contact of sperm head to the vitelline membrane of egg, it gets activated to resume and complete its meiosis too. With this, it gives out the second polar body. The germinal vesicle oogenesis into female pronucleus. At this stage, it is the true ovum or fusion of egg and sperm. The coverings of male and female pronuclei degenerate, allowing to chromosomal pairing. This results in the formation of a synkaryon by the process called syngamy or karyogamy. The zygote is thus formed. The proximal centriole received from the sperm helps in formation of the synkaryon spindle and cleavage of cell into two blastomeres. Significance of fertilization. Secondary oocyte completes the process of oogenesis and is transformed into a mature ovum N number. The diploid chromosome number is restored in the zygote by the process of syngamy. The ovum lacks the centrioles necessary for further divisions are received from the sperm during fertilization. Fertilization involves fusion of male and female gametes from the two parents. It results in variations which are significant to evolution. Sex of the offspring is determined. What we learned today: fertilization or syngamy, mechanism of fertilization, movement of sperm towards egg, entry of sperm into the egg, acrosome reaction, activation of ovum, fusion of egg and sperm, significance of fertilization. Thanks for watching the video. Kindly like, share and subscribe.